We had studied about the transcription unit, which has the structural gene flanked between the promoter and terminator. Now we are going to see how transcription takes place, where we are going to get RNA. When we talk about RNA, before we start with the process that will be depicted in this series of diagrams, we are going to see how many types of RNAs are there. There are 3 types of RNA. One is transfer RNA. The second one is ribosomal RNA, and the third one is messenger RNA. When we study about transcription over here, we are aiming at achieving this messenger RNA in both prokaryotes and eukaryotes, of course. This tRNA has a role to play in bringing amino acids. You know, ultimately the proteins which are to be formed for gene expression, they are nothing but polypeptides. They are formed by amino acids. Amino acids would be brought by transfer RNA. So, it is going to identify the amino acids and bring the amino acids according to the gene's signal, and gene would signal it in the form of codons, the triplets that we had talked about. Then we have ribosomal RNA. It has both structural and catalytic properties for the protein synthesis. Okay, it is responsible for catalysis and structural association that is required, because ribosomal RNA would be present in association with ribosomes. And what are ribosomes? Ribosomes are the cell organelles which act as site of protein synthesis. And lastly, we have the messenger RNA that will have the RNA transcript. Instead, it would have all the information that DNA wants to be expressed as a protein. So, that lies in mRNA. Now, we are going to study about the formation of mRNA in both prokaryotes and eukaryotes, and by the end of this lesson you will be done with what happens in prokaryotes. What happens additionally in eukaryotes would be studied in next lesson. For now, do keep in mind whatever we are studying in this lesson happens in both prokaryotes and eukaryotes. Okay, the additional ones would be covered later on. Now, we are going to see the formation of mRNA, messenger RNA. This process of formation of RNA is a polymerization reaction only, where instead of deoxynucleotide triphosphates as we had in the case of the DNA replication, here we will be having only nucleotide triphosphates which would come and polymerize and form the RNA. Now, the enzyme which is responsible for formation of messenger RNA, or simply I would say RNA, is RNA polymerase. Like we had DNA polymerase 1, we had DNA polymerase 2, over here we have RNA polymerase, and RNA polymerase 2 is the one which plays an important role. We will come to it whether it is 2 or 3 later on, but for now you have to remember that it is the RNA polymerase which single-handedly carries out the process of transcription. Now, transcription has to take place in that part of DNA which acts as transcription unit. It has a promoter region and a terminator region. Whatever lies between this part, promoter and terminator, would be converted into the RNA. The information would be taken, it would be coded, and RNA would be formed with the assistance of this RNA polymerase. Now, this RNA polymerase brings those nucleotide triphosphates, it polymerizes the reaction, and we have in our hand the mRNAs. Also, as I told you, it single-handedly does everything. So, how is it possible that a polymerase enzyme is able to identify the promoter sequence as well? As in the earlier cases, we had origin of replications which were identified by primers. Over here, we don't have primers. So, who comes to the assistance of this RNA polymerase and helps in identify the promoter sequence? That you have to remember that there is a sigma factor that combines with RNA polymerase. Okay, supposedly this is the RNA polymerase, or I would represent it. This is the sigma factor. Inside we are having the RNA polymerase. They both combine and identify the promoter sequence. Once the promoter sequence has been identified, Of course, the helix uncoiling has to be done. The helix gets uncoiled and RNA polymerase enters inside and extends the chain forward. Okay, this is RNA polymerase. It has extended the formation of chain. RNA is being produced like this. RNA is attached. This is RNA that is being produced. This is RNA polymerase that is uncoiling and forming the RNA transcript. This identifies the promoter region, it enters it, it uncoils, it starts forming the mRNA. So this point is point of initiation where RNA polymerase combines with sigma factor, identifies the promoter sequence of the transcriptional unit and starts the transcription. The transcription moves further and further through the structural gene, and the second point is known as elongation. As you can see, it would move further and further and the RNA would become elongated. At one stage, this RNA polymerase is elongated. It comes in contact with the terminator sequence. Here, who comes to the rescue of RNA polymerase? That is rho factor. Okay, sigma and later on rho factor comes to its assistance and the RNA polymerase gets separated from the RNA transcript. So, at the third stage, which is of termination, what happens is the RNA polymerase, that is elongating RNA by adding nucleotide triphosphates, it stops it because the rho factor combines with RNA polymerase. It helps in detaching the RNA, that is RNA strand that has been formed, from RNA polymerase, and in a way transcription comes to a halt. Okay, this is what you have to remember. Now, as I told you, the process still here is similar in prokaryotes and eukaryotes. This is similar process. Now, in prokaryotes, there is no compartmentalization. Nucleus is not separate from cytosol. Okay, whatever is there, it is present in the cytosol. So, as soon as this RNA is being formed, there is no problematic concept of forming this RNA, converting it into messenger RNA, and the protein formation, that is protein synthesis, known as translation, may start simultaneously. So, in the case of prokaryotes, transcription is quite simple. It does not need additional processes like splicing of the introns. Now, two new terms, splicing and introns. What are they? That would be covered in next lesson. Let us revise what we have studied. We have studied the three stages that are present in the process of transcription. Translation is in next lesson. There are three stages of transcription which occur in the transcription unit, that is nothing but structural gene which forms the RNA flanked between promoter and terminator. From the template strand, RNA polymerase notes down the sequences, brings the complementary nucleotide triphosphates and elongates the RNA. When it reaches the terminator sequence, rho factor comes to its assistance and detaches the RNA from RNA polymerase, and RNA polymerase is in turn ready for another cycle of transcription. So, this is all you have to remember in transcription. I hope you have understood it well.